Like everybody else, when I was a child, I was curious. I was curious about everything that I see around me. I was curious about plants, the flowers. I was curious about animals. And later in life, when I started studying science, then I went deeper into nature. And one of the things that interested me, that fascinated me, was how nature solves problems. And I noticed that nature has two ways of solving problems, two extreme ways. And these two ways are actually diametrically opposite each other. In one instance, nature uses so many resources. It is wasteful. And in some instances, it uses as few resources as possible. It is parsimonious. And that is what I want to discuss today. Wastefulness, I use as an example our immune system. And for thriftiness or parsimony, I use as an example the simple viruses. There's a basic law in nature, and that is anything that can be eaten will be eaten. And as you can see, we are good eating. We have muscles, we have many things in us that many animals love to eat. We don't have to worry about lions and tigers anymore, at least not in the Philippines. But here we still have to worry about crocodiles and sharks. But the creatures that we really have to worry about are the smaller creatures, the parasites, the bacteria, the viruses that continually try to get into us and eat us. Examples are head lice, kuto, surot, and many other insects, and especially mosquitoes. Not only do mosquitoes feed on our blood, but also they introduce into us some parasites, like the malarial parasite, like dengue virus. So our main line of defense against pathogens is our skin. As long as our skin is intact, then bacteria and viruses cannot go in. However, we have openings. We have to breathe. We have to eat. And when we breathe in viruses. That's how we get the cold virus. That's how we get influenza. And when we eat, especially if we eat insufficiently cooked food or insufficiently washed vegetables, then we could be getting into our system some parasites, worms, bacteria, and so on. We have mechanical means of getting rid of germs and other foreign substances. If we smell something that is unpleasant, we sneeze, we cough. When we get into our system something that we don't like, we vomit. And in addition to this mechanical means or mechanical means of defense, we have chemical means of defense. Our saliva has antimicrobial properties. Our tears have antimicrobial properties. And then, of course, the parasites will have to survive the acid in our stomach. The acid in our stomach has a pH of 1.5, and very few creatures can survive that much acidity. But many pathogens somehow manage to get through those barriers, and when they do, then they have to contend with our immune system. Our immune system is made up of molecules and cells. And my favorite molecule, of course, are the antibodies, because I've worked on antibodies for many, many years. And then for cells, there are cells that produce the antibodies, there are cells that help those cells produce antibodies, and there are cells that kill parasites and other pathogens outright on their own. And so what I'm going to be talking about now would be antibodies. Our immune system is quite robust and dynamic. As you can see from this slide, we have gazillions of immune cells and even more gazillions. Sorry for the use of this infantile term, but we have many antibody molecules, many cells. And not only that, we produce new cells every second, 1 million new cells every second. Even antibody molecules would be 10 to the billion every second is what is produced by our immune system. Why so many? Well, that's because we have to prepare ourselves for any eventuality. And the molecules that we produce, like the antibody molecules that we produce, are exquisitely specific for the foreign substances or antigens against which they are produced. And not only are they very specific, but they also bind to these molecules with very high affinity. The example that I used to describe the specificity of antibodies is the blood group substances. We are of 3 basic types, blood types. We could be type A, type B, or type O. And a few would be type AB. There are other blood group substances like the rhesus factor, and we could be positive in regard to the rhesus factor, so we could be O positive, A negative, etc., etc. About half of Filipinos are of type O. About a quarter are type A, another quarter are of type B, and a few percent are type AB. Now, what's the difference between type A, type B, and type O? There are antigens, and the antigens of type A are different from the antigens of type B, and so on. And if we are type A, then we have antibodies to type B. If we are type B, then we have antibodies to type A. If we are type O, like most Filipinos are, we have antibodies to both type A and type B. So, what are these blood group substances that distinguish between the different types, or different blood types? The blood group substances, the ABO type substances, are glycoproteins. And they're very similar to each other, except in the terminal sugar. In type B blood, the terminal sugar is a galactose. In type A, it's N-acetylgalactosamine. In type O, there is no terminal sugar. So what's the difference between galactose and N-acetylgalactosamine? There's only one minor difference between those two molecules. And at one position, galactose has an hydroxyl group, and in the same position, in N-acetylgalactosamine, that position is occupied by an acetylated amino group. It's a very small structural difference, but antibodies are actually able to distinguish between these two very similar molecules. That's how specific antibodies are. Actually, antibodies can distinguish between enantiomers, left-handed and right-handed molecules. Now, what happens when a patient is given the wrong type blood. If a patient is given as little as 30 ml of the wrong type blood, then the antibodies of that patient would destroy those foreign cells, and the destruction, the lysis of those foreign cells could kill the patient. 30 ml of the wrong type blood could kill a patient. So what I want to do now is to try to examine, to try to explain the structural basis for this exquisite specificity and high binding affinity of antibodies to their specific antigens. And later, after we have studied the structural basis for this specificity, then we will look at how the genetic mechanisms whereby we develop these many different antibodies with high affinity. Here is a schematic representation of the antibody molecule. It's usually represented as a Y, and at the ends of the arms are the binding sites of the antibodies, and then the stem or the body of the antibody is used for something else. And here are some data regarding human antibodies. There are 5 different types, and they have different purposes. Like IgM is the first antibody type that is produced in infants, and also even in adults. The first antibody type is of the IgM type. IgG is the most common that is used, and later we will see why that is so. And IgE is the one that is involved in allergy. And for some reason, we have antibodies that produce allergy. Anyway, there are the antibody molecules are made up of 2 types of chains. The heavy chain, because it is longer, and the light chain, which is about half the size of a heavy chain. And there are 2 light chain types, kappa and lambda. And the thing is, any light chain may associate with any heavy chain. And if we look closely at the antibody molecule, then we find that both light and heavy chains actually have domains, and the N-terminal domains of both light and heavy are variable, and then the rest of the antibody molecule is the same in antibodies of the same type. Variable means it is different from antibody to antibody. So here is an actual structure of an antibody molecule, and there are 3 main fragments. The ones that are involved, the arms that are involved in binding to the antigen, are called the FAB, or fragment that is antigen binding. And then the FC is the one that is responsible for a variety of functions. And here is the schematic representation of the antibody molecule, where you have the variable domains and the constant domains being shown on the right. So we have the variable domains, but despite the differences in sequence of the variable domains, they have a very similar structure. And actually, all antibody domains have the same basic tertiary structure or fold, and it is called the immunoglobulin fold. The immunoglobulin fold is very strong, and the strength of the immunoglobulin fold is due to the fact that it is mainly made up of beta-pleated sheets. And beta-pleated sheets, because of the many hydrogen bonds between the strands, is actually a very strong structure. It is very strong, And a testament to his strength is that you can change the length, the sequence of the loops that join the strands. You can even cut those inter-strand loops and the structure will not collapse. And so the immunoglobulin fold was actually chosen by nature to be the fold of many molecules that are on the surface of our tissues, our cells, because many of the, or a lot of molecules in our blood and tissue fluids are actually enzymes. And those enzymes are, many of those enzymes are proteases, and they would cleave protein sequences. And again, as a testament to this choice of nature for the immunoglobulin fold as a basic structure in many of its molecules, is the fact that the immunoglobulin domain is found to be present in many, many molecules. Actually, the most number of molecules in the human genome. Here are examples of proteins with the immunoglobulin fold. They have the same basic beta sheet structure, and then, as you can see, the loop structures vary in length and in the nature. Some are helical, some are ordinary loops, etc. This is one of my favorite quotes. Nature is always experimenting. But then when she stumbles on something that is very useful, like a strong basic structure, she uses it over and over again. There are other examples of nature choosing or keeping a basic fold. So we still have to look at the structural basis of the binding, high binding affinity and exquisite specificity of antibodies. Many antibody-antigen structures have now been determined. More than 100 are now available. And in all cases, the part of the antibody that is observed to be involved in the binding to antigen is the N-terminal tip of the FAB. The ends, the tips of the binding arms of the antibody molecule. And here is an example of one of those FAB structures and its specific antigen. What I have done here is actually to lift the antigen about 14 angstroms above the binding surface of the antibody molecule. And I also show the surfaces using dots of the antibody and the surface, the part of the antigen that the antibody binds to. And looking closely at these surfaces, one finds that for every bump in one surface, there is a corresponding depression in the other. In fact, there is a very close shape complementarity between the antibody binding site and the antigen to which it's the part of the antigen to which it binds. Not only is there shape complementarity, but there's also a complementarity in the physical-chemical properties of those two surfaces. Whenever there's a charged group in one finds an oppositely charged group in the other. And that is the basis for the specificity of antibody-antigen interactions. Now, how does nature generate so much diversity? Well, if you look at the sequences of the variable domains, one finds that there are actually regions which are hypervariable. There are 3 hypervariable regions in the heavy chain. There are 3 hypervariable regions in the light chain. And it was hypothesized very early on that these hypervariable regions are actually the ones that produced that constitute the antigen binding site. And it's found out that it was true. Now, these hypervariable regions not only vary considerably in sequence, but also in the number of residues. And because they constitute a continuous surface, then one could actually show the CDR surface in every antibody structure that has been done. And here I have compiled the CDR surfaces of the first 49 antibody structures that had been determined. And as you can see, there is variation. Some surfaces are actually flat. Some have grooves. Some have bumps. And you can imagine that the structure of the antigens to which they bind would have the corresponding complementary surface. A flat antigen binding site would be complementary to a flat antigen, the flat part of an antigen, and so on, and so on. And so the variability in those CDR regions, CDRs, are actually the cause or the basis for the diversity of antigen binding specificities. So that is the structural basis for the diversity and the affinity of antibodies. Now, what is the genetic basis for this variability? When the human genome was sequenced, then they noticed that the light chain gene, the gene that that is responsible for the light chain, is actually made up of several gene segments. And that is true for both the lambda type light chain and the kappa type light chain. And that's also true for the heavy chain. The variable domain of the light chain is formed from a variable gene and a J gene, or a joining gene. And there are roughly 30 variable genes and 4 J genes in the lambda type. And any of the variable genes, any of the V genes, can associate with any of the 4 J genes in the lambda type. That is true also of the kappa type. There are about 40 V genes and 5 J genes, and any of the 4 V genes of the kappa type can associate with any of the 5 J genes. It has been found that the variable domains of antibodies is actually formed by the combination of V genes and joining genes. And in the human genome, there are roughly 30 V genes of the lambda type and 4 J genes of the lambda type. And any of the 30 V lambda genes can associate with any of the 4 lambda J genes. And that is true also of the kappa type light chain. There are roughly 40 V genes of the kappa type, and though any of those 40 can associate with any of the 5 J genes. That is actually even more complicated in the heavy chain case. There are roughly 40 heavy chain V genes, and there are 6 heavy chain J genes. But in addition, there are 25 diversity genes, or D genes. And again, any of the 40 heavy chain V genes can associate with any, and not just one, maybe even several of the D genes, and that combination can then associate with any of the 6 J genes of the heavy type. And so the total diversity, if you compute from these many different possibilities, total diversity of the antibodies that could be produced is 5 times 10 to the 13, a huge number. And there are other affinities and other specificities could be produced. And one is by somatic hypermutation. That is, the cells that produce the antibodies could actually be activated by helper T cells. And when they are activated, then they mutate, and the daughter B cells could have different sequences. And after several cycles, one finds that there are mutations in the CDR regions of these V genes, and somatic hypermutation could also result in new affinities and new specificities. And so you produce a lot of different antibody molecules, diverse, high affinity. But in our lifetime, we probably encounter only a few million antigens, different antigens. So why do we have to have 10 to the 13 different antibodies? It is so wasteful. But then we cannot know what we will encounter. And so to be prepared, we produce many millions upon millions of different antibodies just in case we encounter an antigen that our antibodies could bind to. We cannot know. So we are being very wasteful. Wastefulness in nature is not only noted in the case of the immune system. We also note that nature is wasteful in the case of, for instance, reproduction. In reproduction, in human reproduction, the female of our species offers one egg, one cell, and the male, in every attempt at first fertilization, offers several 100 million of his own cells, when only one is actually needed. So why is there so much waste? That's because nature makes sure that the one male cell that fertilizes the female cell is actually the best, the one that can overcome the barriers that the female system offers. And so in this case, only the best will propagate the species. And this is true also in plants, in pollination. When plants try to pollinate, in the instance of pollination, plants release billions upon billions of pollen to make sure that the chance of fertilization is actually improved. So in the case of the immune system, wastefulness results in the best fit. The best fit would be would ensure the survival of the individual. In the case of fertilization, wastefulness ensures the survival or increases the chances of the best molecule, the best cell being produced. So in the case of fertilization, it is the survival of the species that nature is trying to ensure.